Welcome to Classique tutorial number 6. In this episode, we will take a closer look at the main output of the analysis, the cluster view table. The cluster view table contains 4 subtabs: info, gene clusters, protein groups and filtering. They can be displayed or hidden using the buttons in the top bar. The info subtab has 2 options, info and general info. General Info subtab provides an overview of the parameters that were set for the analysis. In the Gene Cluster subtab, you can click on any gene arrow within the displayed cluster. When you do so, the Info subtab will populate with detailed information from GenBank about the selected gene or protein and its homologs. This includes direct links to GenBank, the protein sequence, and functional annotations of both the encoded protein and its homologs. The Gene Cluster subtab contains the list of species in which the identified gene cluster is present. Here you can show and hide further subtabs. We will describe them step by step. You can easily adjust the size of each window. In the cluster view, your marker proteins are all highlighted in red. Other proteins in their surroundings are represented by simple arrows. Primarily, the size of the arrow relates to the size of the protein, but you can switch to the fixed size and back. When you click on any arrow, Classique will automatically highlight all homologous proteins in the cluster view. Use the right mouse handle to open the next menu. Select Set Color to change the color of all arrows representing homologous protein group. Select Align to order the cluster view. Right-click and Customize enables to rename the protein group. You can also set color of the arrow, text font, etc. Classique automatically groups clusters according to their level of similarity. To find rare or unique proteins represented in the clusters, press /highlight rare button. The protein groups that are only found in one cluster are colored dark gray, those that are found in fewer than 20% of all displayed clusters are colored light gray. You can sort clusters by shared protein groups, it is a default option, or by taxon name. When you click on the taxon name, relevant information from GenBank will appear in the subtab Info. You can also search for a specific taxon or group of taxa, for example the one which the marker proteins originate from. Clearing the search window will return you back. If you are interested in a specific protein or protein group, you can mark any arrow of the group and check the info window. Besides basic information from GenBank, you might find useful list of all related proteins of the protein group and list of all GenBank annotations of proteins within this group. In Protein subtab you can sort proteins in descending or ascending order, check the annotation and number of unique and total counts within the cluster file. You can also search for a specific protein using or protein ID or any key from annotation. To learn more about Protein Groups subtab, See our tutorial number 7 Protein Groups, Basic. Filter window enables selection of clusters according to presence or absence of any combination of proteins. To learn more about filtering, see our tutorial number 8. In any time you can rename your cluster view. You can also create another cluster view after colocalization with different parameters. Finally, you can save your project to .clp file and come back to it later. To start from .clp file, see our tutorial number 2. Thank you for using Classiq.